Hello everybody, welcome. We are talking about different mobile genetic elements because we already have several lections, meetings about bacteriophages. We talked about plasmids, and now I decided to make like a short bonus lecture about mobile genetic elements, because when we talk about all these structures like plasmids, bacteriophages, there is no actually strict border between these elements, because we can isolate a plasmid and later we will understand that it was a prophage, a lysogenic state of bacteriophage, or we can find a bacteriophage that acts like a plasmid, or actually we can deal with the phage that acts like a huge transposon. And if you do not know what is a transposon, so you need to come here and we will talk about mobile genetic elements. We will talk about their types. There are a lot of them actually. I chose the main, the core, the most meaningful for the understanding of idea of mobile genetic elements, and we're going to try to sort of classify them. So if we're talking about mobile genetic elements, you understand that we are going to talk about some sort of elements of genome. They are rather small, not that big as chromosomes. They are much, much smaller. And the main thing is that they are mobile. They can move. They can move within genome of one cell. They can move through different cells like bacterial cells. They can move through different cells in microorganisms. They can move between kingdoms, like from bacterial cells to plant cells to cells of humans or animals. So these elements are mobile. They play a very important role in the horizontal gene transfer, in the horizontal evolution. They cause different mutations. They cause genome changes. They can silence genes. They can bring the genes back to life. So this is actually their role in nature, and they are sort of a moving force of this horizontal gene transfer. So let's begin. Let's start with something simple and move to something more complicated. The first type of elements I would like to talk about, just say a few words, are IS elements or insertion sequences. As you can tell from the name, these are sequences that insert. So they insert themselves into larger molecules. Plasmid DNA, phage DNA, bacterial chromosome. I will focus on bacteria and phages and plasmids. I will not talk about eukaryotic cells, but we do also have in our cells all these types of mobile genetic elements. So, insertion sequences. This type of mobile genetic elements is called selfish because the only genes they carry are needed for their needs, only for themselves. So they do not bring any new properties to the cells, to phages. They only carry the gene that encodes its enzyme transposase that helps them to be mobile, and inverted repeats on their ends, these sequences that also help them to move within the genome that this transposase can recognize. Well, they have their certain functions. We won't focus on them. So the idea is that these insertion sequences are rather small elements, and they can insert into the bigger genome molecules. When they do it, they can do it like in the middle of the gene, and we have the gene, it is functioning. If we put something inside, the whole molecule stays whole, but the gene is broken into parts and it doesn't work anymore, and the cell doesn't receive the product of this gene anymore. So the gene is silenced. If this element leaves, the gene will be full again. But if this element stays, the gene is silenced and it's not working any longer. And actually it can cause some sort of disease, like in the human organisms, animal organisms. It can cause the death of bacterial cells. So the consequences can be different. So usually these mobile genetic elements, they use two strategies to move within genome. The first one is when they build in, they stay for a while, and then they can leave and move to the other place. So one copy of these mobile genetic element travels through the genome. The second variant is that it builds in, replicates its copy. The first element stays on the place where it was inserted, and its copy moves around the genome. So, like making copies, the copies move through the genome. The copies cause those mutations, insertions, and changes in genome. So, insertion sequences. Sometimes they're called simple transposons. Let's talk about transposons. Another type of mobile genetic elements, these are transposons. Some of them can be, as you can see, compound. So there is a sort of classification. Transposons have their names like TN5, TN10, TN9. I worked with this transposon. I used it to mark a cryptic plasmid, and so that it was possible to build a restriction map of this plasmid. And if you watched videos about plasmids, you understand what I'm talking about. If not, well, you have the chance to understand. So, transposons. Why they are called compound? Because transposons are not as selfish as IS elements. They also carry some genes that can be useful for the cell, for example. They carry antibiotic-resistant genes. They can carry resistance to one antibiotic or several antibiotics. But anyway, if we talk about the cell that is resistant to antibiotic, probably this resistance was brought by the transposon. And somewhere in the genome of this cell, a transposon is, going around, walking around the genome. You can see that these compound transposons have insertion sequences in their structure. So IS elements, yes, IS elements, they are smaller, and here they are on the endings of our transposons. They also help the transposon to build into the genome, to move within genome, and they are already part of these transposons. So, insertion sequences are mobile genetic elements. Transposons also are mobile genetic elements, only a little bit complicated. They are larger in size, and they already carry some genes that can be useful already for the cell. So, these transposons, they can insert, built into the genome of plasmid, bacteriophage, bacterial genome. So it doesn't matter, they build into some bigger molecules. And we already talked about plasmids, and as I already said, plasmids also can carry antibiotic-resistant genes. Sometimes these parts of plasmids are transposons. So transposons are parts of plasmids. Let's repeat it. Insertion sequences, small. Transposons can be compound using insertion sequences. Plasmids can have in their structure transposons. And what is very important to understand about mobile genetic elements, there are no exact borders, like strict borders, like this is the plasmid, this is the bacteriophage. Some phages act like big complicated transposons. Some phages act like plasmids. Some plasmids reveal to be lysogenic state of bacteriophage, so prophages. And now we're going to talk about them also. So here is their example of a bacteriophage mu, that actually acts like a very big, complicated transposon. This phage, mu phage, received its name, mu, means mutations, because its life cycle causes a lot of mutations. So this huge phage is like one big transposon. When it enters the cell, it acts like any other phage. Its DNA enters the cell, and then it incorporates into the chromosome. And by the way, it can stay within the chromosome in the state of lysogeny. So this cell is lysogenic with this quiet phage. It sits quietly. But if this phage starts somehow its lytic cycle, or it is induced, this phage uses replicative transposition. It means that it not just simply replicates itself, but its copies, they also insert the genome. They also enter the genome of the cell. They also cause mutations. Sometimes these phages that enter the genome of the cell, they can take some genes. Well, occasionally it's like a mistake, but anyway, they can take some genes from this genome and these genes, they appear into phage particles and they move to any other cell. So this is a horizontal gene transfer performed by the phage that acts like a big transposon. So you can see that everything is connected. Phage acts like transposon, and it can take some genes out of genome and move these genes horizontally. So everything is connected in the world of mobile genetic elements on this molecular level. Some phages, as I already said, can act like plasmids. You cannot tell whether this is a plasmid or a phage unless you make some really, some serious research or simple research. But anyway, you have to carry the research out to find out is it a plasmid or is it a bacteriophage. It is true for phage P1, for example. Well, you know the classic way for phage replication within the cell. The phage enters the cell, it replicates its DNA, the proteins are synthesized within the cell, so the expression takes place. And when we have phage parts assembled, they break down the cell and go out. So new generation of phages is ready. If we talk about lysogenic state, the phage, the genome of the phage enters the cell, then it enters the DNA, and it stays quietly within the cell as a prophage, so it's part of the genome. And if we talk about, this border state, like what is it, a plasmid or is it a phage? The phage can enter the cell and stay within the cell as a, for example, megaplasmid or medium-sized plasmid. It cannot be a very small plasmid because it carries a lot of genes that are needed for its life cycle. But anyway, it's middle-sized or megaplasmid. So a plasmid, probably low copy number plasmid. Again, it depends on the size of it and its properties. It doesn't need a lot of copies. So low copy number plasmid, but actually it's a phage. And this state, it doesn't replicate, it doesn't act anyway. If it has, for example, like some transposons, and they can move from the genome of the phage to the genome of the cell. We're going to go back to this a little bit later. But one more thing about this interesting system: when this phage enters the cell and stays like a plasmid, it turns on toxin-antitoxin system that helps the phage to maintain itself in the cell line. When this cell will divide, two daughter cells must acquire one copy of this phage plasmid, at least one copy, because if the daughter cell won't take the copy, won't receive the copy, it will die. This phage encodes toxin-antitoxin system. If the cell has the plasmid, it has both toxin and antitoxin, and it stays alive. But if the cell, the daughter cell, won't get the copy of the phage plasmid, the plasmid phage, won't get the copy, it will die because it contains only toxin. And the antitoxin is already ruined, and we need this DNA to get more antitoxin, but without this plasmid, there won't be antitoxin. So the cell, without this phage, without this plasmid, will simply die. And this is the way how this bacteriophage maintains itself in the cell line, so that the cells will divide and won't lose him. So he lives his quiet life without his physical form, but it stays within the cell and continues his life. So, about horizontal gene transfer, mobile genetic elements, Their relationship is very complicated. Insertion sequences, they carry genes that are needed for transposons to move within the genome. Transposons are compound, they have those insertion sequences in their structure. These transposons can be part of plasmid, they can be part of phage genome. Phages and plasmids, they can look similar within the cell, and we cannot tell from the first sight whether this is a plasmid or this is a phage, so we need to bring a research. And all these mobile genetic elements, they take part in the horizontal gene transfer, like the evolutionary force, the force that changes our genomes, causes sometimes problems, sometimes brings new properties. So how it happens? The simplest explanation. Here we have the cell, here we have the plasmid. When the plasmid somehow enters the cell, well, we can also imagine that this is not a plasmid, this is like a phage entering the cell. So anyway, we have some foreign DNA entering the cell, and it contains a transposon in its structure. For example, this transposon also contains resistance to some sort of antibiotic, or the ability to produce some toxins, or the I don't know, I don't remember what else it can be, the ability to produce some substances that help the bacteria to kill other bacteria and stay alive. So there can be a lot of properties. So this is the gene on the transposon, and they are this new foreign DNA and this chromosome, they are both within one cell, and this small transposon can jump onto the chromosome. We already do not need this element, so our transposon is already jumping around the genome, and it changed the properties of the cell, it changed somehow genes of the cell, so causing mutations, causing changes in genome by entering it. So this is actually the way how it happens. We can have a transposon that is on the chromosome. It can jump from the chromosome on the plasmid, or it can jump from the bacteriophage genome to the genome of the cell, or from the bacteriophage it can jump to the plasmid. The plasmid move horizontally to other bacterial cells. So this is the horizontal gene transfer caused by mobile genetic elements. Mobile genetic elements are connected with each other. They can be parts of each other. Some bacteriophages are more like big transposons, so they act like transposons, but anyway, they are bacteriophages. Some phages act like plasmids. Some plasmids carry transposons, some transposons carry insertion sequences, so everything is connected in this molecular genetics, in this molecular world, and it causes a moving force, a moving power of changes in our genomes, of the movement of genes that is very important for bacterial evolution at least, and it can also influence our lives, and it can help us to create biotechnological vectors that will change the genomes for the better. Like if we talk about some broken genes, we can use these mobile genetic elements to bring normal genes to the cells. So these technologies, they actually at this moment have no borders. It's like one of the leading fields in science, studying space and studying biology. These are very, very interesting, very, very important fields in science right now. So I hope I just tried to make it a little bit clearer for you, the information about mobile genetic elements. Well, I think that anyway it will be useful for you if you want to understand the connections between different parts of this genome of bacteria, because today the tendency is to creating the not watching just a piece of the picture, but creating the whole picture. For example, scientists who work with plasmids today, they talk about plasmidome. It's like the whole pool of the plasmids present in some sort of ecological niche. Like, for example, we can take a human being and talk about the bacteria that are present within the body of the human being, and we talk about the plasmids that are present within those bacteria. So this is the plasmidome of the human body. And we can talk about the pool of the genes that is concentrated in this plasmidome. So probably sometimes really genes that influence life of large organisms, they can be not part of the chromosome, but part of the plasmidome, situated on plasmids. And all these things, they are not separated, but they are connected. They create the whole picture that helps us to understand this molecular, this small world, and make big things in biotechnology using these small molecular biological approaches with mobile genetic elements. So thank you for your attention, and goodbye.